Thank you. So this work was done at Radboud UMC during my PhD. And I would like to go back. So we were hearing um, a lot of great talks, but I would want to go back to the structure of the EHMT1 and functions. And then we'll talk about two different types of variants that we have analyzed. So here is the linear 1D structure of the EHMT1 protein. And so you can see that the N-terminal part is in white and it's largely disordered, so it does not have any 3D structure. We don't know why it's important, whether it's important, so it's just there. Then at the C-term end, we have the preset and set domains, and they are the catalytic domains that we know of, that they perform this enzymatic methylation function. A lot of people forget, but actually this preset and set domain is also important for binding between EHMT1 and EHMT2. And actually, this only complex of EHMT1 and EHMT2 performs this methylation activity. And lastly, there is also anchoring repeat domain. And it is actually a reader, so it brings the EHMT1 and EHMT2 complex to the histone so that they can methylate it. So they are all important. And if we're talking about the known spectrum of the pathogenic variants causing Kleefscher syndrome, so the most obvious, the first known is the deletion of the 9q34, including EHMT1 gene. If we look in our in-house database of the variants that were found at Radboud UMC, so we see that most of those variants are frame shifts and nonsense, so loss of function variants spread throughout the gene. And if we look on the published and discovered pathogenic missense variants. So they're shown below, and they are only 3. So when we started at the moment, it was really, loss of function predominant gene. But with the current application of the exome sequencing, so everybody's finding VUSs in all kind of gene, including EHMT1, and Professor Kleefscher receives a lot of emails, so what do you think about this variant, and what do you think about that? And so at one point, there were so many emails that we decided, we gotta collect and look on this systematically. And then we looked in our in-house database in different NDD projects, and also all of those emails we collected, and we ended up with 37 individuals with 33 different EHMT1 missense or in-frame variants. And so we wanted to classify each of these variants and to understand which are pathogenic and which are not. And for this, we looked on the clinical features, on the effects on the protein 3D structure, also on the inheritance and in silico tools as in ACMG guidelines. But then, of course, this would be only predictions. So we needed some functional test. So one of such was DNA methylation signature, and this was done in collaboration with Bekim Sadikovic in Canada, and also in vitro functional assays done by Professor Yuichi Shinkai and Ayumi Yamada in Japan. So for those who doesn't know about the EpiSign, so it's a relatively new test, and it uses DNA methylation in blood to classify the variants. So here you see the PCA plot for Kleefscher syndrome analysis, and in green you see the healthy controls, and blue are the Kleefscher patients. And you see that there are clear separation between these two groups. And when we tested some of the VUSs, some of them were negative and some of them were positive. And you can see that the negative are in orange, they are together with controls, and the red ones are with the Kleefscher. So that's how we know that they are positive. Very similar analysis is also just shown visually differently is on heat map and clustering, and there's also the machine learning SVM behind this that provides yes or no answer, mostly for positive or not. And so we were lucky enough to collect a lot of from DNA from all of these cases. And for the nice thing is that you can run a lot of samples in parallel, and you need also blood-derived DNA that can be stored, that was stored in a freezer even for 10, 20 years. It works fine. And so the results that we got, that some of them were positive and some of them negative on the signature. And you can see that when we correlated the results of the signature, of the episignature with all the tests that we looked on the 3D and clinical stuff, 30 out of 33 variants had the same classification in all of the points that we used. So 11 were benign, and they are shown in black, and 19 were pathogenic. So we started with 3, and we ended up with 19 that we know are pathogenic. And if we look now, what is the proportion of the protein-altering variants as the cause of the Kreiser syndrome? So when we looked at the RedBud UMC exome sequencing data from almost 10 years, that was collected by Ariana, the data. So we see that about 20% of all of the pathogenic variants are due to the missense variants. And similarly, in another DD cohort from DDD study, so they did not have CNV data, so this was already excluded from the inclusion. But basically, similarly, from the point mutations, de novo point mutations, 25% are protein-altering variants. And then there were 3 challenging ones that we thought are pathogenic, and the signature said it's benign, it's not Kreiser. So yeah, we were struggling a bit. And so I'll try to explain why. So this is the 3D model of the set and preset domains, and it's in cyan color. And in red, you can see the variant that causes Kreiser syndrome. And so they are located either here binding zinc or somewhere deep in the core of the set domain. So if there is any mutation, the whole protein is just destroyed and it's not functional, it's a pure mess. But then we had these 3 variants that are in purple, and they are located in the active core of the set domain. So here is the, with the cyan, it's a cofactor, so it's SAH, and here with orange, it's histone 3 tail. And you can see that this histone 3 tail, this is lysine 9 that goes directly into this active center. And so the mutations that we had here that were classified negatively in purple, they actually, we predicted they would disrupt the active core, but not the set domain in total. So these were only predictions. We needed some functional data to back it up. And this is where Professor Yuichi Shinkai agreed to help us. And so they performed some functional tests. So they tested binding capability between EHMT1 and EHMT2. They have analyzed enzymatic activity of EHMT1, and they also tested the ability to bind to the histone 3 lysine 9 protein fragments. And because it was too laborious to do it, all of these tests for all of the variants, we cherry-picked those that we wanted from different parts of the proteins. So we have selected 3 from the ankyrin repeat domain. We have selected 2 pathogenic that are classically pathogenic, but we know 100% causing Kreiser syndrome in the preset and set domains. We have selected 1 in black that's benign as a negative control, and of course these 3 that were discordant. So I will start with ankyrin repeat domain. So this is in green. Does anybody recognize this picture? So then you didn't look at your gift bags. So we received 1 parcel. For the next year, we probably should receive the set domain, yes, so to make a puzzle. But this is the ankyrin repeat domain structure, and we use it not only to make fancy gifts, but also to analyze the protein variants. And here are a couple of examples, but I want to, for example, stress this one. So this is tryptophan 912 mutation to arginine, and you can see that it's actually located in a surface that really binds the histone 3 tail. So any mutation at this point would likely disrupt the binding to the histone 3 tail. And when this was measured in in vitro system, then you can see here the binding capability for in percentage for the wild type, for unmethylated histone 3 tail, and mono, di, and trimethylated histone 3 tails. And you can see that for all of these variants that were tested, but especially this tryptophan 912 position, there was a decrease in binding capability. So our predictions were correct, and this variant indeed lost its ability to bind to the histone 3 tail, at least in partial. And then going back to the other variants and the other tests, we also tested, as I mentioned, the histone 3 lysine 9 demethylation activity and EHM2 binding ability. So here on top are the 3 discordant set domain variants, 2 set domains variants that causes Kreiser syndrome, and the 3 ankyrin repeat domain variants. So as expected, ANKIN repeat domain does not affect methylation activity or binding capability because it has different function. Then if we look on those two variants that causes Clavius syndrome in SET domain, they disrupt EHMT2 binding ability and enzymatic activity. Because as I mentioned, they disrupt the whole domain. It's a mess in there. But then these three variants that we have tested, so actually two of them turned out to indeed disrupt the enzymatic activity, but the overall change of the protein was so mild that it was still able to bind to EHMT2. And then again, this protein had intact ANKIN repeat domain, so it was able to provide some of the functions. And for one of the variants, it seems we were wrong. We thought it's pathogenic, but in all of the tests that we performed, it showed normal wild-type activity. So we're not sure what's going on in there. So, and then the question is, how can EHMT1 lose enzymatic activity but do not have Clavius syndrome episignature? Because that's what we believe, that EHMT1, the main function is this enzymatic. So going back to the functions, so one of the first main functions is EHMT1 binds to EHMT2 using this SET domain. Next, using the ANKIN repeat domain, it brings the EHMT2 to the histone 3 tail. And then actually what is shown in vitro that most of the methylation activity is done by EHMT2 and likely not EHMT1. So we think that EHMT1 is not as important for the histone methylation. And it's also known that EHMT1 does have methylation activity, but maybe for the non-histone proteins. For these non-histone proteins, we know it occurs. We don't know, is it important or not, why it's occurring, so it's a big black box. And just as a summary, so if you disrupt these first two functions, you have classical Clavius syndrome, and if you lose only enzymatic activity, you have something that's not Clavius syndrome, or not typical, at least, Clavius syndrome. So just a summary about the protein-altering variants. I want to stress that the pathogenic missense and in-frame indels are common, but previously was underappreciated cause for the Clavius syndrome. Then protein-altering variants in ANKIN repeat domain disrupting only histone 3 binding is already sufficient to cause Clavius syndrome clinically and also on a DNA methylation level. And similarly, protein-altering variants in SET domain that disrupt EHMT2 binding, but not only enzymatic activity, also is sufficient to cause classical Clavius syndrome. So this was one part of the story. We have another cryptical cases that we don't know what's going on about. So, and this is one of the example about the N-terminal variants. So, kind reminder that most of the pathogenic variants that were known before causing Clavius syndrome was the deletion of the whole gene. And then another part of the history was published in 2011 that Professor Clavius discovered 3 individuals with de novo deletions that are shown in here, that all of them had typical features of Clavius syndrome, but the deletion itself did not affect the coding part of the gene. And then, so yeah, I was at school at that moment still, so, but as I understand, while looking more into the conservation, turned out that there were actually 2 extra exons that was previously unknown. And this prompted the update of the transcript from 3 to 4 versions, and that's how EHMT1 got extra 31 amino acids. So there is again a problem with the old papers that you have the different position of the variants because of this extra piece. And yeah, and now when this was known that this deletion actually deleted the first exon, and it makes sense that they were causing Clavius syndrome. And then it's all again started with an email request and with a single patient, because there was a family, a daughter with a frameshift mutation that was inherited from completely unaffected mother. So mother did not have any symptoms. And the question was, what do you think about it? And so, yeah, this was at first for us, but we actually looked more, and we at the same time received in, I think, 2 months, 2 different emails with a similar content. And in total, we were able to find 5 families or individuals that have truncating variant, frameshift variant in the first 2 exons. And so I think this one is very big, that there is 4 individuals with EHMT1 frameshift variant. It's actually a deletion of the second exon. And the only affected is a daughter, but she also has a pathogenic ASXL3 variant. So she has another cause for her symptoms, and everyone else was unaffected. And for these two cases, we do not know the inheritance, so we do not know whether it's de novo or not, but we do know that all of these people that have these variants, they are either unaffected or very mildly affected. And when we tested on the episignature, then we tested actually all of those family members as well when available. And for 4 out of 5 families, it turned out to be negative. So as saying, they do not have DNA methylation signature as Kleefstra syndrome. And one case, the first one that's had also XXX syndrome, was inconclusive. So what does inconclusive means? So inconclusive means that machine learning algorithms say it's benign, it's not Kleefstra syndrome, but if you look on the data, then you see some differences. So here, again, the same plot, green one are healthy controls, blue ones are the Kleefstra syndrome, and the red ones are typical truncating Kleefstra syndrome causing variants. And these N-terminal variants that I mentioned before, they are shown in yellow, and you see that they are all clustering with the controls, except maybe for this one. It's, I would say, in a limbo state somewhere in between. And if you look on the heat map, here it is again, it's more with the Kleefstra cases rather than with the controls, and other are really with controls clustering in there. So this was the inconclusive case, that AI says it's benign, and the visualization says, So, yeah, this is the response that we get. Yeah, exactly. So what we believe happens here is that we actually know that there is an alternative start codon that can be used in third exon. And so if you have the mutation in the first 2 exons frameshift, then the translation can be started with an alternative start, starting third exon, and you actually likely cells produce the shorter version of the protein. The thing we do not know whether these variants are actually hypomorphic, pathogenic variants with incomplete penetrance, or they are just benign. We had only 1 photos from all of those cases and this patient had inconclusive features, inconclusive DNA methylation, and facial features were similar to Kleefstra syndromes, but for others, we unfortunately were not able to receive facial photos to compare. And the problem is that we do not know whether these first 31 amino acids are important for the protein function. We know that there are happening some post-translational modifications. So, for example, the alanine gets N-acetylated, but yeah, whether it's important for protein to lose it or not, we have no idea. And that's why we also have no idea whether these variants are pathogenic or not. So take-home messages for me, at least from this project, was that if we comprehensively characterize variants found among affected individuals, we're able to pinpoint main protein or gene functions, as well as look on the disease mechanisms that are important for causing a condition. We're also able to identify subphenotypes, and yeah, what's dear to my heart is already provide recommendations which variants are likely to be pathogenic and which are not, so the correct diagnosis could be made. And yeah, I'm young and naive, and I thought that EHMT1 was already studied for decades. We should know so much about it, but then when you end up with some questions, yeah, you still don't know too much about it, and you still, yeah, it still lacks the interpretation for the variants. And of course, this was a job of a lot of people, but of course, Kiecka Kleefstra driven this whole process, and also we're working with Ariana now to put it all in a manuscript so that it would be out. And of course, the London and Japanese group help a lot. And thank you very much for your attention.
Thanks so much, Dimitris, for presenting this. It's indeed very challenging still, all these variants, and to interpret and to counsel this correctly.
Thank you very much. I think this is going to be a great tool for diagnosis, especially very, challenging patients. So my question is, on those mutations that affect the activity of EHMT1, is it possible that EHMT2 is somehow covering for that lack of methylation?
Yeah, that's what we think, that it's not even covering, but I think that EHMT1 is not that important for methylation in the first place. And that's, if you lose only methylation activity, at least for the histone methylation, I think EHMT1 is doing its job as it should.
Yeah. Okay. Very nice. Congratulations.
Thanks.
Yeah, very nice, Dimitris. So, but you say methylation is not important, but you mean methylation by EHMT1.
Methylation by EHMT1 on the histone 3.
Yeah,
Yeah.
yeah. I think this is an important.
Yeah,
difference.
exactly.
Yeah,
Yes.
because I think the change in methylation histone methylation can still be a readout or biomarker that tells a lot. So we talked about it yesterday. So if you look at gene expression throughout all the cell types, you do actually see quite some difference in EHMT1 versus EHMT2 expression. I did not do a systematic analysis, but could we, looking at your model, I think this might be interesting to see. There are some cell types where you have more EHMT1 than EHMT2, and looking at your model, it seems that EHMT1 is the limiting factor. So, is there anything we can learn from looking at the expression of EHMT2 and EHMT1 in different cell types?
Yeah, so this was your suggestion that we should do that, look on different cell types, the proportion. The problem is that a lot of these histone modifiers have non-enzymatic function, and that we don't know about. So that's, I would want to bring back to the basic science people, we should learn more about what EHMT does, and then we could have better understanding why it's at some tissue it's expressed more than EHMT2 without this enzymatic activity. Because I really think that there is extra to this. There is a whole N-terminal part that we don't know what it's doing, but it's there for a reason, right? So yeah, I think there is extra function that we just are unaware currently. And that's why cells need EHMT1 only at some places.
Do we have any other questions?
About this N-terminal domain, it's fascinating indeed. Is it also possible that these early frame shifts lead to just a reduced amount of protein that is still functional, so that after all the N-terminal domain would have not such a dramatic function, but just the concept of an early termination reduces total protein amount?
We did not test this, so we did not have patient cell to look for the Western blot, the amount of proteins. I do not have ever, so then only part of the transcripts would get eradicated right from the cell, but then still you have an early frame shift, you gotta have somewhere the alternative start. So there's still maybe a couple of mechanisms, yeah, indeed, but.
Okay, any other ones? Okay, thank you, Dimitris.